I wanted to tell you today about one of the projects in my lab, which actually started at the Hadassah Medical Center with a family that came for genetic counseling. So 2 parents and their firstborn who has a developmental delay and a diagnosis of autism. And when they came, they wanted to know what was the actual problem so they can deal with it in their next pregnancies. And so what the hospital could offer them is what we have right now, and it's sequencing just a fraction of our genome. And so 1% of our genome, which is 3 billion nucleotide long, actually codes for proteins. And so the DNA is our book of how to make proteins. And proteins are actually the building blocks that builds our body. And 1% of it, we actually know what it does and that it builds protein. The hospital sequenced 1% of this kid's genome. And what they found was a mutation in a system that exists in each of our cells that does editing. The way to get from the DNA to the proteins is actually through RNA. And so the DNA codes for RNA that later translates to a protein. And human beings, we only have 20,000 genes, but we have 500,000 proteins. So how do you take 20,000 genes and actually make 500,000 proteins? And so the way to do it is called editing. And so we can take each of our genes and edit it differently to make different proteins. This kid, it has a mutation in the editing system, but he has a phenotype, or he has a problem only in his brain. We know that the editing is very important, and when it changes, it can cause autism. But what we don't know is which of the genes editing is important. So once we understand how editing is different in this kid, so we can actually try to find a drug to change it, and maybe try to address some of the symptoms that this kid has that makes his life harder, and try to make it better.